He was a great administrator, and he loved us like his children.
Very great, very great administrator.
Yes.
Very great.
Yeah.
There are no words to describe him, and discipline was immaculate during that time. Ragging at its peak, followed by discipline. Yeah.
So we must have ragged you also. Okay.
So Surinder sir, should we start?
Yes, let's start.
Yeah. Devashish, over to you.
Okay. Thank you, sir. So first of all, good evening to one and all present here. So as you all know, the saying that nothing changes if nothing changes. Well, if nothing changes, we stay the same. We don't grow. We don't evolve. We don't get better, and that's not going to work, and not for neither for you and not even for the world. We need some positive change. We need new ideas every day. We need progress, and for all these, first and foremost, we need the innovative brains. Hello everyone, very good evening to one and all present here. I Devashish, on behalf of Student Alumni Relations Cell, BITMAA, take this opportunity to welcome Surinder sir, Anirudh sir, Vishal sir, and all other those who have spared their time for this webinar. Now, I would request Vishal sir to take the event further. Sir, please.
Thank you, Devashish. And first of all, I'm really thankful to BITMAA North America to bring this faculty webinar series. It is almost after 4 months of gap that we are again having this webinar series started. The first in this series from Dr. Anirudh from Johns Hopkins University. After this, we have another webinar next month, and I'm thankful to Surinder sir and BITMAA North America team to arrange all this for us at this particular moment when we all require knowledge sharing. Without further ado, I would request Surinder sir, please welcome Anirudh sir and introduce him to the audience.
Thank you, Vishal. It's a great privilege to welcome Dr. Singh. He is a BIT alumni, graduate of 2001 in polymer engineering, and then a master's from IIT Bombay and a PhD from University of Akron, Ohio. And he has been assistant professor at Johns Hopkins University since 2015, and his interest is in finding clinically relevant simple solutions to complex health problems through translational scientific and engineering approaches. And one of his main goal of research is regenerative urology, and that's what basically his topic of speech is today is challenging solutions to regenerating the urinary bladder. And as I was telling earlier, that Dr. Singh loves BIT so much that in June, when Dr. Manna was here in US, he traveled 200 miles to meet him and offer his services for the ISRE program which BITMAA has, and for the faculty webinar and the faculty exchange program. And also he offered his services to be a mentor to BIT students. It's indeed great honor to have someone as an alumni like that. And I wish all alumni had that same spirit as Dr. Singh has. So without further ado, I will welcome Dr. Singh. Let's give him a big hand. Thank you, Dr. Singh. Please start.
Thank you so much. Thank you so much. It was so kind of you talking about me a little bit. We know each other since last few months. And I was honored to be with you and with Dr. Manna and other people. I wanted to, before I begin, I wanted to ask you, can you see my screen?
Yes.
Yes.
So it says, okay, awesome. Normally I use Zoom, so this is different format, but hopefully it is working out. Okay, awesome. If anyone has question, they can ask in between or after the presentation. And I wanted to tell especially students that you can ask any questions and don't be afraid asking anything. I'm not going to judge anything. I want to make sure that we can utilize this time at max. All right, so let's begin. I'm going to talk about reconstructive urology, mostly bladder tissue engineering. Urinary bladder, everybody goes to pee, and without emptying our bladder, we won't be able to survive very well. So you can imagine the urinary system, the first picture in the left side. It is with kidney, ureter, and bladder, and the passage of urine happens through urethra. And the bladder is like a rubber balloon. You can imagine a balloon, slowly it fills with urine, and then when it reaches to a certain pressure, it collapses, letting the urine out. And it happens more or less 1 hour, 1.5 hours, depending on the bladder volume and the age of the person. I always say that our heart is beating every other second, but urinary bladder is doing the same kind of thing for like in 1 hour or so. And without that, we won't be able to lead a good life. Oftentimes, at least for some percentage of people, something happens and there is a cancer development. You can see that tiny bump here in the picture, second picture, that would lead to bladder cancer. And in that case, we need to take out that particular tissue or the whole bladder to treat the patient. Another example is some unfortunate kids develop this extrophy condition where the spine is not well formed and the bladder goes backward and there's a bulge coming out of the back of the child. This gets treated as well. Unfortunately, if the child is born with this particular situation, they survive for like 25 to 30 years, but their quality of life is in general poor. Fortunately, nowadays, there's a technique which can solve this issue when the fetus is in the womb. So surgery in the womb can get rid of this issue, but people or children who are born with this disease, they have to go through multiple surgeries and need to have a bladder. And I also want to show you some bladder histology just to have an idea. This particular picture is showing you the top.
Dr. Anirudh,
yeah.
Dr. Anirudh, sorry, your slides are not changing.
Yeah, hang on. I'm just
on the first slide.
Yeah, I'm just going to do that. I'm just introducing it because I don't think everybody knows about urology here.
Okay.
Just giving some background and then I will move on. And then the rightmost slide is called histology slide. People who go biopsy, who do biopsy. The top surface is urothelial cells. It's called superficial layer. And why I'm saying that this is the most important layer in urinary bladder because it keeps the urine in place and not allow it to seep through the muscles. If urine seep through the muscles, there would be a lot of problems. And sometimes people die as well. So this is the most important layer in bladder. And this is the most tightest layer. It won't let the urine go through it. And that is needed in urinary bladder. Saying so, let's move forward. And I would start a little bit telling people about me. How did I start this journey? I was a polymer scientist or engineer, and then why urology or tissue engineering, right? So you can see that I started Bachelor of Engineering. You are able to see my full screen. Let's do this. Bachelor of Engineering in Polymer Engineering. Now the program is called Chemical Engineering, I think, and it has Polymer Engineering as a sub-branch. And of course, being a teenager at that time or transitioning from teenager to adulthood, we had the most wonderful time in BITMAA-NA. I was a very studious guy. I can only say that I invested 4 years of my life only studying. And sometimes I regret that I should have enjoyed that life a little bit more. And my friends used to say that just have some fun. And I was so focused because the competition was so cutthroat and I don't come from a very rich family. So I had I had only limited resources. The only thing what I knew is study hard and succeed. And that paid off. I went to IIT Delhi for my master's. I was fortunate enough to become All India ranked second in GATE exam. And I went to Indian Institute of Technology for Polymer Science and Technology degree. And while doing that, I went to University of Aachen, RWTH we call it, in Germany as a DAD scholar. And then I applied for PhD. Now you would see my PhD is in polymer chemistry. I was trained as an engineer, but became a chemist because I knew a lot of things about making things, engineering things, but I really wanted to make molecules, how the polymer is made in the lab. So I started from scratch, working with vacuum lines, Slenk line, high-tech chemistry, all those things, and learned from scratch and got a degree in PhD from one of the best in polymer science. And then I went for a job in General Electric. Now it is called Sabik Innovative Plastics. As a product developer, I was working on polycarbonates. Polycarbonates with the plastic lenses we have, glasses or helmets, or you can imagine cars, the tail light, where the light is coming from, that plastic is basically polycarbonate. And I'm proud to say that I was in the team who made the first iPhone casing, iPhone 3, I think, the casing of iPhone. I would say that at that time, I was not very fond of industrial job. Somehow I felt that I could contribute more toward academia side. So I switched back to academia. I went for a postdoctoral fellowship in Johns Hopkins University in Biomedical Engineering department. Again, it happened to be the #1 department in USA, and it is still maintaining that rank. So very fortunate to go to the top schools everywhere. And in postdoctoral fellowship, I worked on cartilage. Cartilage is the tissue in the knee. People who have osteoarthritis problems, knee problems, mostly the cartilage is not working properly. And I also work on eyes, ocular tissue engineering, regenerating cornea, develop eye drops with hyaluronic acid. And I did postdoc for almost 4.5 years, 5 years. And then I was promoted to become a faculty in Hopkins. Normally, we have to become an instructor for 1 year. And based on the performance, you will be promoted to assistant professor. And this is how I came to urology. People were looking for regenerating bladder. And I was being a tissue engineer, I was hired to do that. And in 2015, I joined as an assistant professor, and since then I have been there. So it has been quite a bit of journey, a lot of studies, a lot of hard work, also luck, a lot of great mentorship in between. And this is why I am here. And I also want to return back to new students, guide them, mentor them to, do it right ways and succeed in their life. And we are also very thankful to people like Dr. Sharda, who has been helping and bringing these things to BIT community. At that time, when I was there was nothing like this. So if we have something like this now, we should always take advantage of it. So let's go back to tissue engineering in urology. I have kept the slides simple enough so that most of the people can understand. Hopefully it is simple to understand. Again, bladder cancer is a terrible disease. It basically impacts a lot of lakhs of people. And every year around 70,000 people are coming with bladder cancers. And out of them, 13,000 people are dying. The overall cost is tremendous. It's almost like $12 billion, I think, in recent figures. The problem with bladder cancer is, a lot of people smoke. And I don't feel any hesitation to say that if somebody smokes, stop smoking, because this is one of the major reasons for bladder cancer. And it's not a nice disease to have in later life. And the kids who have this extra trophy, this is very unfortunate. Fortunately, the number of cases with the kids is going down every year because of the genetic tools that we have. We can see that if there is an issue with the fetus, and they can take care of it earlier. So this disease has been going down, so that is a good news, but still a lot of cases come in Hopkins, and Hopkins is like #1 treating pediatric patients. So saying so, if somebody has this kind of bladder conditions, what is the gold standard? What is the current surgical procedure? So you can see the first part of this slide. It's called bladder augmentation. What they do is they cut a small part of the intestine from the same patient, and they use that as a tissue to cover the bladder area which was cancerous. If the cancer has spread everywhere, they take out the whole bladder and create a new bladder from this intestine itself. So this is one way to create another system for urine to retain and pass out from the body. And this is a gold standard. The simpler model of that is creating a tube as a passage from the small intestine. You can see in the second figure, a new urinary conduit. You can see the last image here. There's a tiny tube which comes out of the abdomen area, and it is connected with ureter. So all the urine which is coming drop by drop from the kidney, it will eventually go out of the body, and there's a pouch next to the abdomen. Now, urinary conduit is mostly done. Most people prefer this method because it is most for it's easy to handle in day-to-day life. However, for long terms, it creates some major problems. There are conditions where urine leakages happen, and since intestine is not created for urine, it is created for food absorption and secretion of enzymes. There's always problems with mucus. Intestine is full of mucus, and you have to keep cleaning it. Urine can pass through the tube. And there's a formation of stones, and eventually kidney fails because of the back pressure. So the quality of life is relatively poor.
Dr. Anirudh, you are muted. Dr. Anirudh, you are mute.
Hello?
Yeah, it's okay now. I would request the audience, if you people are not interested, please don't disturb the speaker. You are not forced to be here. Please don't disturb the speaker. Yeah, sir, continue.
So you were able to hear my earlier things, right? Or what happened? Why it was muted?
Just 30 seconds ago, you got muted.
Okay, all right. So I was talking about the current methodology for treating bladder cancer or dysfunctional bladder. So they use patient's small intestine itself and make a passage for urine to pass out of the body. Now, the good news is it saves the patients, but the quality of life after surgery is very poor. And that is why surgeons and scientists have been looking for alternative strategy. So what are the alternative strategy? Let's see. Alternative strategy is with biomaterials. You use a biomaterial which is biocompatible, and then let the bladder regrow on that biomaterial. So biomaterial act as a scaffold where cells can go and make the bladder tissue itself. Now in biomaterials, there are several types of materials. You can say synthetic polymer based, polymer, biopolymer, or natural polymer, and extracellular matrix derived scaffolds. Now this is where the polymer knowledge is coming. This is a training what we gone through our degrees. undergraduate and master's degree. Now, out of these three categories, there are different materials, and many materials have been tried. And out of these biomaterials, ECM derived, basically what they take out some tissues from pigs or bovine, and then they decellularize it. They get rid of the cells from that, and whatever tissue is left, they use that as a template or scaffold. Now, out of several biomaterials, ECM derived biomaterial, they are very advantageous, and they are clinically used for many purposes, not necessarily for bladder purpose, but for many purposes, because there are a lot of biological factors present in that tissue. They help for wound healing or tissue regeneration. However, with any material that you work with, any tissue engineering, there is some issue, there are some plus minuses things. And so far what we have found that the tissue that we use or material that we use for bladder reconstruction, it shrinks when it is implanted in the body, fibrosis happen, the tissue quality becomes more stiff. It is not the same that we would like to have. And sometimes the mechanical properties of the regenerated tissue is not enough as the host site will have. So there are good things, bad things. We have not reached to the point that we can say that we have a successful biomaterial for any particular purpose. Now, I was talking to you about urinary bladder. You can imagine as a balloon, which is when empty, it's very tiny, but when it fills up with urine, it balloons up. Okay, and then it collapses again, going back to the rest position. So expansion and contraction is the nature of urinary bladder. Now I want you to think. We have different organs and tissues in our body. They are made of collagen. Bone is collagen and calcium. Bladder is made of collagen and elastin, and skin is collagen and elastin. But all of these tissues have different mechanical properties, right? Bladder, which is like a balloon, stretches quite a bit at a minimal force, but the bone is otherwise it is very stiff. So if we want to solve a problem within urinary bladder, we need to create a system which would work like bladder. This is kind of hypothesis. This is obvious to proceed further. So when you go back and check how the bladder works, what is happening at the molecular level, you can imagine a spring. You can imagine a ribbon. A ribbon is folded, or spring is in helical shape. When it stretches, it expands, and when there's a signal that hey, there is enough expansion, let it go, it collapses, goes back. So the ribbon opens up easily, and then it goes back. This process is basically modulated by collagen 3. The particular collagen protein is called collagen 3. You can see from the left image, the right side of the leftmost image, the ribbon opens up and expands and goes back. So this is the relaxation and expansion of the bladder, and which is present in our urinary bladder. Unfortunately, so far we have not been able to create microfibers or nanofibers which would mimic this physical phenomena and then create scaffold out of it. So we have always been looking for other ways to create a system which is made of collagen but expands and contracts easily. Saying so, I told you earlier that urinary bladder has a job to keep the urine isolated, and it should not leak, and it should not go into the bloodstream. So blood-urine barrier. How it happens? This is the most tightest barrier in our body system. You can see the left image here. What you see with boundaries, these are cells, and they are so tightly packed. There's no room for urine to go through anywhere. And if you zoom in through the structure of the cells, you will see they are like fractal objects kind of structure. These are made of proteins. They are called uroplakeins, and they are regularly spaced. And these all structural things play important roles for urine barrier. So far, what people have done, they take a biomaterial and use it as a template or scaffold to create bladder, but we have to consider what is the mechanical property, how easy to stretch that material. At the same time, would that material stop or resist urine leakage? Because if we don't do that, the implants fail. Saying so, there have been possibilities. In 1960s, 70s in Japan, they started from plastic molds, literally taking a plastic mold and creating a bladder out of it in human body. And then they used gelatin sponge, and they also used paper. It's a special paper called Japanese paper. The good news was they showed that in human, this Japanese paper were able to create the whole bladder tissue, partial tissue. And the patient was alive and he was doing fine. Unfortunately, they did not have a lot of patients and they did not do any follow-up studies, but it showed the possibility that maybe we would be able to regenerate bladder. In 90s to 2000, when decellularized extracellular matrix, the tissues from animals, they were using that and they showed that we can potentially use these tissues for reconstruction of bladder. So a lot of possibilities, a lot of hope. Recently in 2010, around 2010, there was a company called Tenjian. They used polylactic glycolic acid based scaffold. They made a tube out of it and they seeded some stem cells on it derived from the same patient where it was going to get engrafted. And then they followed up what happens with the wound healing or tissue regeneration. This is for urinary conduit, a conduit which let the urine pass through from the body or pass out of the body. And this is a simpler model than creating the whole bladder. If this works, we can go to the next version, which is creating the whole bladder. And it has complicated system, nerves, neuron signaling, everything is there. So it becomes a next challenge, next level of challenge. So conduit is easier. Making a tube is easier. Unfortunately, we cannot even make a functional tube yet from the cells and biomaterials. Biology is very complex, but we are learning slowly. Now, the material that they have used or the clinical trials that they went through, this material was a stiff material. It takes a lot of force to stretch it. Now, bladder or bladder related organs or tissues, they are very elastic. They are very rubbery like. How can you use a stiff material to make that happen? When I saw that, I was like, why they're using this material? You can see the stress strain curve. The first one is the stiff material. It takes a lot of force to just pull a little bit of this material. If you use a compliant material, a rubbery material, it takes minimal force to stretch it. And after certain stretching, it resists for stretching. For example, if our bladder is full, it does not have to keep ballooning more and more because otherwise you will have trouble. So it starts resisting that I cannot hold more urine. You have to let it go. And then there is a neural signal that says that, okay, let the urine go. So when we go to pee and then bladder collapses. So the second graph here, second curve here is more showing more resemblance toward bladder or bladder-like tissues. We want material which shows this behavior, not this behavior. So we have two things. Can we create a collagen-based material which is compliant? Another thing is, can we introduce urine barrier to that material? So how did we achieve that? This is very interesting story. In our laboratory, we were working to create a coating on our material which will resist urine, which won't let urea. Urea is the major component of urine and which causes the denaturation of proteins and all the problems happen because of that. So we wanted to create a coating with some wax-like material. We found that a material called dodecynyl succinic anhydride or any higher version of it with higher methylene groups. So what I am writing here is C18. C18 means there are number of CH2 groups or total number of carbons you can imagine. It's like a fatty long aliphatic chain, fatty acid long aliphatic chains. When you react this with amine groups present in collagen, it creates a system which partially works as cross-linking sites. Now I want to tell you a little bit about cross-linking here. In general, whatever scaffolds are used for tissue engineering, they are chemically cross-linked. So they create static points in the system to introduce more strength to it. But what we did was transiently cross-linking. And what it does is you create cross-linking sites which are temporary, which can be disturbed, but by thermodynamic principles, it comes back and forms the cross-linking again. So basically, you are introducing a system in the collagen 3D network with some aliphatic chains, which have their own preference toward another aliphatic chains, and they form like a micelle system. So you stretch it. Since these micelles are not super strong, they would let the stretching happen. But when you leave it, they like to rearrange themselves into the same resting position. And that's how the stretching happens and contraction happens. So these hydrophobic groups are introduced into the collagen system to bring compliance into the system. Now this happened not because we wanted to do that. This happened because we wanted to do it. We wanted to make a coating of aliphatic chains because they are hydrophobic in nature, so they will resist anything in the water and water. Okay. Now the good thing is it works with collagen. It works with gelatin. Gelatin is denatured version of collagen. You basically heat the collagen, it becomes gelatin. It loses its strength. And decellularize extracellular matrices, the tissues which come from animals. So one simple system can go into different matrices and potentially solve our challenges of compliance and urine barrier. Let's see the stress strain curve. You can see figure A, B, and C. There are different kinds of decellularized extracellular matrices. The green curve shows before modification with this aliphatic chain, and the blue one shows after the modification. The nature of the curve itself shows that how the stiff material, if you see the B, which is more prominent here, graph B, before modification, this animal-derived tissue has high stiffness. You can see the sharp slope here in the green curve. And then when you modify it, it drops and becomes like a more compliant, more rubbery-like material. And you can see the water contact angle increased. You see nice formation of water droplets on the modified material. That means that it is able to resist water more than unmodified ones. The good thing was we could also create any sizes and any shapes from the gelatin derived material. You can make a tube out of it, or you can make it like a balloon out of it, and it would hold the water. So what is the beautiful thing here? It is a very simple experiment. Why simplicity is needed? Because for clinical translation, for commercial purposes, you need simplicity. You don't want to make things very complicated. It's a simple thing. It is able to make collagen and collagen-based materials, including DECMs, into a compliant material. Great. And it also resists urine. How? You can see that this is an experiment conducted with chambers where the material is put in between and the urea is added, urea solution is added on top of it, and let that solution pass through the material. How much material has passed, that determines how much urea resistance it is. And we found this number here with the arrow is the lowest in the modified material compared to the unmodified material, and commercially and compared to a commercially used DECM material, which is small intestine submucosa. So just by modifying, you are able to resist urea significantly. So we are creating urine barrier, we are creating compliance in the material. Now going back and using this material into the mice bladders and seeing what happens to the tissue regeneration. So left side picture is showing different layers of the tissue. The top layer is called urothelium, the middle layer is called submucosa, and the bottom layer is called detrusor muscle, the muscle which gives its strength. The top layer is involved in urine resistivity and a lot of signaling. Submucosa helps to make it thinner by compression or extension, and detrusor gives it strength of the bladder. So when we say after 8 weeks of the tissue regeneration with this biomaterial, what happens to cystectomy control? Basically a surgery which is done for control purposes to compare. And then we have control biomaterial and modified biomaterial. The modified biomaterial is that we just made compliant and urine resistive. And histologically, if you see detrusor muscle, DM, LP is lamina propria or submucosa, and UR is urothelium. All these images look very similar. That means that our material after 8 weeks was able to regenerate different parts of the bladder. And you would see that the control material had urothelium which was very thick compared to the modified biomaterial. However, this urothelium is still thicker than the normal healthy bladder of the mice. So it is a positive indication that our material is working. It is able to regenerate every layer. Now let's see, does it really function the way it should function? Does it expand and contract? So what we do is called myography. In myography, you use this regenerated tissue and clamp it in between two fixtures, and there's a transducer connected to it, and a particular chemical, it's called carbachol, is used to stimulate the muscle. So basically it makes it contract and relax, contract and relax. So how much contraction happens, we measure it through this curve in shown here with respect to concentration of the chemical used. And you will see that the white bar here is for control, healthy bladder. The gray one is for cystectomy control or the material that is not modified. And then the black one is with the modified material. So you can see that there is a nice increase in the contraction with respect to concentration of the chemical used. However, the biomaterial is never able to reach to the healthy samples. It is behaving in similar manner as it should, but the contraction is not as good as the normal healthy ones. Now, you would ask me the question, well, we introduce right set of properties in biomaterials, even then we are not able to get functional properties. Yes, that is the reason why we do research, right? So now what is happening? Why it is not behaving the way it should? So what is beyond biomaterials? This is the molecular question that we are coming, molecular mechanism. So we have a very good biomaterial now, but let's see what are the events happening at the molecular level, cellular level. What kinds of cells are coming? What kinds of cells are disappearing throughout this regeneration process? Is there a particular cell subtype that is missing? Or is there a particular cell subtype that we need to get rid of? So for that, to determine that, we work with single cell RNA sequencing. What kind of gene expression is happening at what stage of tissue regeneration? Just a brief introduction to people who are listening to this talk. What we do is we collect bladder from the mice, and then we isolate live cells from those bladders, and then we run gene expression sequencing as through Illumina sequencing machine, and we create plots that what are the genes, what are the components of muscle layers, epithelium, lamina propria that are showing up in the system. And then whatever we find, we validate through different techniques that these results are correct. So we did that. Let's see. So we did that, and we plot a curve, two-dimensional plot, and which shows different groups, different colors in the leftmost side. You can see that, and these are called, some of them are fibroblast, urothelial superficial layer, basal layer, endothelial cells, smooth muscle cells, neuron, immune cells. Everything can be seen from this graph, and we can know how much percentage of these cells are present in the group. We did the same thing for the regenerated tissue, and we profiled them, that what kind of cells we are getting. So we get more or less same subgroups of cells, and it overall gives you a picture that what is happening after 8 weeks of regeneration. We also plot a variance plot. Basically, it shows what genes are going up. The red ones are going up and the blue ones are going up compared to the healthy ones. So if you have a baseline, that what is the gene expression in the healthy one compared to that in regenerated bladder, whether some genes are going up, or meaning that they are more expressed and some of them are going down. Now you can see that there are different genes going up and down, and this would tell you that what are the events that are happening there, and is there any significant differences. If there is a significant difference, we can address that by using those cells from where these gene expressions are happening, or we can do gene therapy to basically modulate that. And the same story goes with urothelium, the urine barrier, because it is most important layer. And we do find some differences. You can see the healthy tissues in the left side, and the right side is regenerated tissue. And we can also plot what cells followed what trail, what cells became what. And that is like a pseudo time trajectory. With time, what cells are becoming what. And we can literally chalk out the path of the cells taken, and we can find out differences in the regenerated versus healthy. Now the good thing is, by this method we can find out what are the common genes in the healthy and regenerated ones, because to compare anything we need common ground, and we need common genes expressed in different layers of urothelium, submucosa, detrusor, which would tell you these genes are coming from these layers, and we were able to do that. You can see from the yellow highlighted ones. Now we wanted to see how these differences are panning out in different phases of tissue regeneration. When you have a wound healing process, first inflammation happen, a lot of different cells comes together and start building that tissue, and then tissue granulation, tissue gets mature and they have some functional properties. So it's like from day 1 of surgery to day 60 of surgery, there are a lot of events that are happening, and each event would have different cells coming, going, different mechanisms. So we wanted to understand the whole spectrum of it to make sure that we can find some differences and address those to make the bladder functional the way it should be functioning. And in that process, we also studies what are the signaling pathways that are impacting the whole process. Now you can imagine that there are so many mice we have to use to understand this time series. So there's a better way to do the same experiment. This came out recently. It's called a spatial transcriptome. What we do is you take slides or sections. You can see the leftmost side, the picture in the leftmost side. It's like a biopsy punch, and you make thin slices of it and use those slices to do the RNA profiling directly on the slice. It saves a lot of mice, the life of a lot of, we have to be very concerned of the lives what we are using. So it minimizes the number of mice. It gives us tremendous flexibility in conducting large scale experiments, and we get the same answers as we would get from single cell sequencing. In fact, it gives you more information about from where, what location the RNA is coming more or going down. So we are doing this experiment, and how does this help? I will tell you. For example, you see this tiny, orifice here. This is with urothelium, and then the bottom one with yellow one is detrusor muscle bundles. So first we need to define the regions, area of interest, that these are detrusor muscle or urothelium, and for that we do histology, and then we do RNA sequencing, and then overlap it, saying that okay, this region is urothelium, this region is detrusor muscle, and these are the genes responsible in these areas. Once you establish that, you do the whole genome sequencing on the slice itself. And when you do the whole genome sequencing on the slice itself, you would literally get from what location, from what part of the tissue, what genes are going up or down. Is there any particular subsets which are causing this regenerated bladder tissue to not function as it should be functioning? And this is very powerful. And if we get that whole information, maybe we will be able to make a functional neurourinary conduit or bladder. So how the results would look like? It will be in the form of differentially expressed genes. The plots would give you different cells and number of cells in those whole area, on the whole area, what kind of cells we would have, and then we would plot it in time in heat map. In heat map, we would be able to see healthy versus regenerated. For example, if you see that the purple color is healthy, yellow color is regenerated ones. So you would literally seeing the differences in every compartment of the tissue, that what are the cells, what are the genes that are different. And once you do that, we would have a powerful set of information to move forward. Now, this is kind of a big grant, big study, and we basically make sub aims to complete the study. So first we'll be determining the location and function of unique cell populations, and then what are the differentially gene regulatory pathways involved in regenerated bladder tissue, and what is the molecular mechanism that we will be studying by spatial transcriptome. And this work becomes a PhD work. And this is under progress by Shivang Sharma. He is from IIT Kanpur, and he was involved in some biomaterial development aspect, and he's doing all this sequencing. So a lot of activities is happening with genomics, RNA sequencing, Python coding, and people are using artificial intelligence, machine learning as well. So learning Python has become a necessity now. Just 2 slides more. I might have taken more than what I thought, but I wanted to make sure that people can understand it. So we keep our eyes open. Okay, this is a fascinating creature. This is a fascinating creature called a cookie. Cookie is the sound that they make, and these are male frogs. They use their gular skin or throat area, and they balloon themselves, and they make sound to attract females. This is made of collagen. You can imagine where I'm going now. The frog throat area is made of collagen. The skin is made of collagen. They're able to blow it like a balloon, and it goes back to the normal size. So if nature has given some kind of species which have this capability to balloon collagen, why cannot we use it as a system to know more about making bladder materials? This is what we were doing exactly, and studied the whole mechanism of frog's skin, this particular frog's skin. And we found that if you do the stress strain curve, you don't have to worry about too many graphs here. The green one is a typical stiff material, the frogs that won't be able to balloon their skin, versus the blue and the red ones, which can do it. You can see they are very compliant, meaning that it does not take too much stress to stretch almost 300% of their normal size. So why so? In bladder, we had the helical shape or the ribbon shape material. Here in male E. cocci, they don't have a special feature except the microstructure. The microstructure is very unique. They form a zigzag structure like Z. If you write Z on the paper and you take the ends of the Z and stretch it, they become stretched. They become a straight line with lot of length. That is stretching. And if you leave it back, it becomes Z again. The same mechanism is happening here with this particular frog that is able to balloon their skin. But female ones, they cannot do the same extent. And the control species, other frogs, they cannot do it at all because they have ridges, they have stress points, and they cannot make it like a rubber. So this work was also published. And it was very fascinating that can we use this kind of structures to create new biomaterials. These are different flavors of the work that we have been doing, and we cannot be enough. What do you say that? We cannot have enough gratitude to thank our funding agencies, NIH, R21, and King Khalid from Saudi Arabia's funding, and supervision for my salary, my student salary. In USA, we have to pay our salary from the grants. We have to pay our student salary from the grant. It's quite challenging in that way, but we are very thankful for the funding agencies. And then our collaborators, Eugene Senderov, he's an oncologist, he's a physician for special transcriptome. Andrew Wald, he's also a surgeon. And Biwalakwa, Trinity Biwalakwa, he's a urologist surgeon. And my students, Shivang Sharma, Justin, he was a master student, Sarah, she was also a master student, Christine, you master student. So these students were involved in the project that I have been showing today. And with that, what I would like you to consider a few points to all students and faculties. I would say that we should target high impact problems. Don't do research for the sake of doing research. We should have a very impactful problem. Shoot for the highest. Work on the current topics and new direction. We cannot work on 1940s or 1950s problems. This won't take you far in terms of publication, grants, and whatever. And of course, be open-minded. Oftentimes, as scientists, we think that our ideas are the best ideas. It may be true, but we always be open-minded. Listen to other people and what they have to say. Entertain their different ideas. And this is my favorite. Allow safe mistakes to happen. Accept mistakes. We are normally scared of mistakes. Oh, what would happen? What my PI would say? What my teacher would say? Mistake is a mistake, and most of the research now happening, scientific discoveries that are happening because of mistakes or that did not fit into the logical reasoning, straight logical reasoning. And like what we are doing, Dr. Chadda is doing, Vishal is doing here, help each other, recognize people, whether he is junior to you or senior to you, support each other, give awards, give more awards. Even if it sounds like it's a very tiny thing, but give more awards because it uplifts you. And there is nothing to lose here. And most importantly, be happy and have the entrepreneurial spirit. Don't look for a job. I mean, you have to look for a job, yes, but be a business owner, create opportunities for others. Just have that much confidence and, enough grit to move forward. Don't worry about things, do things. And in last, I would say meditate. I'm a meditator. I'm a teacher of meditation as well. I teach meditation, and this has helped a lot, getting great ideas and being happy. If you do everything and not happy, nothing works. Nothing is worth, right? And I'm open for any questions, especially to students. I would appeal that don't be afraid to ask any questions. Nobody would judge you. Raise your voice and ask questions. And I'm happy to be here and do whatever I can do to guide you, to mentor you for your successful career. With that, I would like the floor to be open for discussion.
Thank you. Thank you very much, Dr. Singh. Excellent presentation and great advice. It was really great. We really enjoyed it. Let's give us a big hand to Dr. Singh.
Any questions, Dr. Anand, Dr. Anupam, Dr. Akhil Sen? For your information, Anirudh sir, they are from the Department of Chemical and Polymer Engineering at our department, our institute. So if you would have any questions, please feel free to ask. Dr. Abhimanyu Dev was there.
Dr. Singh, you very good.
Yeah.
Hello.
Yes.
Hello. Nice. Can you remember me? I'm Dr.
Yes,
Yes. I came in our department long back we met. Actually, I have joined in the department 2004 actually. Before that, I have passed out from the department. So at present, I am heading the department. So we are proud of you because earlier you were the adjunct faculty to our department. So always you are expecting some advice from you. And you know, our department is changing from polymer to now chemical engineering, but still some batches are still working in polymer students are there. And or some old faculty, Professor Sarkar, Professor Goswami, you might have known them. Your teachers, Professor Sarkar teacher, Professor Gautam Sarkar.
Yes, he was there. I think I have met you as well when I came to visit long time back. So namaste to you and everyone in the department. And Sarkar sir was there. Goswami madam was there. All of those people are still there. I'm glad to know that, and I'm very thankful and my gratitude to all of you.
Thank you. And one thing I have to know that your film you are preparing, which solvent you are preparing, or are you adding some plasticizer for that?
No, so most of the things that what we do is water-based chemistry, because in surgery side, in the biomedical side, we try to keep it as simple as possible, and reactions, we choose reactions that work in water only, in buffer. So the reaction that what we did was in water with little bit of DMSO. It's very simple. You just add those and they react in 30 minutes. And then after that, just wash them with several times with the buffer solution, PBS buffer, and then it is ready to go.
But DMSO, is it toxic now that your small amount of it present, then it may be harmful.
So DMSO is not very toxic unless you're using less amount, like maybe 1% of it. And a lot of drugs are used with DMSO. Existing drugs are used in DMSO to basically do some therapeutical thing. So it can be tolerated to a certain extent, not too much. So we make sure that there's no leftover.
Okay, well, thank you, and we are always welcome to the BIT as well as department. We'll be happy to see you again. Okay.
Yes, definitely. I would look forward for that.
Okay, thank you.
Dr. Anirudh, we have our PhD scholar, Mr. Satyajit, who was also our master student at BIT. Basically, he is from Department of Electronics and currently working into a research whereby he's looking for some solutions in early detection of cancer. So, Satyajit, if you want to ask something.
Hello, sir. Good evening.
Hi.
Sir. Yes, sir. We have also interacted previously, Satyajit Mahapatra. On
right.
LinkedIn, we have,
right?
Yes, sir. We have interacted.
Okay.
Now, sir, listening to your lecture for early detection of bladder cancer, can we get some cancer images along with some data like the concentration of calcium, alkaline phosphate, albumin, urine, ketone, these all things. So in order to make a model,
I think I have seen a few studies that do the same thing that you are asking. So you can get those data, the baseline blood content, urine content, and then in disease at different stages. Disease does not happen in a single day. So different stages, like in the beginning, in the moderate time, or the terminal time. And there has been effort to do artificial intelligence based software development that can predict that if you have a particular trend that might lead to bladder cancer. So in that line, sure, people have been looking into it, and I think I should be able to send you one paper that I have in my computer, and you can check it. And you can apply for different diseases. If suppose bladder cancer is there, you can use for prostate, bladder hyperplasia. A lot of people, old people, have prostate gland, they are enlarged, and you can predict that what age they would have this issue, or what you can do about it. So definitely this is an area that you should look into, and everybody should. Actually, I would encourage everyone to learn Python and learn some coding. It's essential nowadays.
thank you very much, sir.
Sure.
Anybody else? Dr. Means we have Dr. Anupam also with us. Anupam, if you would like to ask a question, something.
Thank you, Vishalji. And sir, namaskar and good evening.
Namaste.
Namaste. Yeah, sir, I'm Dr. Anupam Ghosh. I am in Department of Management. Actually, I wanted to know, I have my father who stays with me, and is 75 years old. He has a chronic problem of retention of urine.
Right.
And he has got no prostate. I've made all the tests and USG is done. The problem is that due to retention and all, sometimes blood comes into the urine. Blood comes and sometimes the infection grows, and we give antibiotics, then it reduces and again comes back. So this has been a recurring process. So what is the way out, as a if you can just advise me?
So this is interesting because there's a lot of age thing is involved here. Of course, I'm not a physician, but what I understand is you should get her, exam done about non-invasive bladder cancer or prostate hyperplasia. Sometimes the blood comes there because of infection. So if the infection is recurring, it might lead to some problems in the later on. So you might have to be vigilant about it. So I would say if a physician or surgeon has not said anything about the cancer, then I would go
nothing,
for
sir. Like as of now, the surgeon has said nothing about, but this thing comes again and again, and that's a very recurring problem that he faces.
Recurrent infection is something that we must address, and I'm not sure whether you have, done some conventional therapies, non-allopathic therapies, because if the disease has not materialized yet, we may be able to take care of by our lifestyle, our food style habits, because we ignore those things so much, but it has tremendous impact. Everything, more or less 75% of the diseases are related to stress and our lifestyle. Okay. Again, I'm not a physician. I don't want to recommend you anything, but based on my experience, my loved ones' experience, I take care of anything wrong in my body through my lifestyle, my life habits, my food selection, what I do with me, Ayurvedic, homeopathy, whatever it works for me, right? Food is the first medicine. But if disease has materialized and it needs to be taken care terminally, then you go run to surgeons. That is a different story. But if there's no particular disease that has been diagnosed, then I would say that go traditional way, conventional way, and see, what you can do. For example, again, I'm not recommending, but you should consult a doctor for panchkarma therapy. Those things are very powerful.
Can you recommend, sir, some foods which can keep bladder like urinary infections away. Some typical food.
Right. So right now I don't have that particular thing in my mind, but I can find out something, and then I can send you that. If you can email me your email address, just email me on Anirudha Singh at gmail.com, and we can check out some literature and some things.
So nice of you. Thank you, sir. It's a big honor. So yeah. So I'll definitely get in touch with you. Thank you so much, sir.
Sure.
Vishal sir, thank you.
Any students?
Any students who want to ask something? Devashish, are you there?
Yes, I am there.
Yes, Devashish, if you wish to ask something, because you are from a chemical engineering department.
Yes, sir. I'm from the polymer technology student only.
Okay, good.
Basically, I wanted to know, sir, about the research work that you are doing. So, if you can briefly explain that.
What do you mean?
So basically, what you are doing in this current research about the urinary bladder and all.
So we are waiting for some recent data on a special transcriptome, and we want to process that data and figure out some molecular mechanism. So right now it is about understanding the biology behind the process.
Okay, sir.
Yeah. So I want to say to everyone here, all the students, you are investing your 1.5 hours of time listening to me, right? Why don't you use this and ask or open up with any comments, questions that might help you? There's nothing to worry about it. Just ask anything, any career advice, or about jobs, or what you're thinking about your department, what kind of studies. Yeah, go ahead, Hers.
Yes, sir. I'm from the new branch of data science, which got started in VIT this year. So you, like in your research, you said you're using a lot of data,
Yeah.
And data analytics is something which is used in every field nowadays.
Right.
So, like apart from Python, what are the other tools that the medical field is using so that your research data can be very well understood? You told us about that everyone should learn Python. So, what are the in details in Python as well as the other softwares that you are using for your research?
Mostly Python and R, because all the algorithms that are written to process genomic data, they are either in R or Python. And there's a website called GitHub. GitHub has all the algorithm that people have used to process any data related to what we are doing. And you can literally download those algorithms. Those are open sources, and process that. So the world has become very smooth now, because we can use someone's algorithm to process our data. So I don't think there is anything else needed. Whatever you need to process data in particular way is already in the algorithm. You just need to understand the language and process it. So you need to learn Python and R. So if you know that, then everything else, depending on the project, would come to you. But in general, about analytics, suppose in finance you go, there is another software for that. But those also use more or less Python or R. So I think just by doing so, you are in much better condition.
Okay, sir. Thank you.
Sure. How many of you are opening any companies or startups or willing to do that? Does BITMAA have a particular department or unit that would support those kind of activities? How about fundraising?
Fortunately, I am the faculty advisor of Entrepreneur Cell as well. And we are already coming up with a technology-based incubation within 1 month, for which Government of India has funded us.
Very nice.
Apart from that, Institute Innovation Council is very much progressive in funding student startup. Recently, we had one business event competition whereby we have given very nominal seed amount to just register them as a company and come up with a ideation plan. which they will be presenting in the month of October. So entrepreneurs is one thing which BIT is now nurturing like anything.
Very good. That is very good.
Yeah, and Dr. Anirudh, if you can also become, because already Surinder sir is here, we are looking forward for mentoring program also, whereby we look in various aspects. So one of the aspect of mentoring would be in the area of this entrepreneurship. So we would request you to be part of our ecosystem.
Definitely, I am always there. I might not come to all the meetings because of a young child, but I would make sure that whatever help I can provide, I would do so. Okay. And I think Surinder sir has so many of experience, life experience he can share to, expedite things. I can do whatever I have learned in my life. I think it would help a lot. And I encourage the bottom line is, see, if you are happy, things would come to you. If you're not happy, things would be harder to come to you. So the bottom line is, I always encourage people to be genuinely happy, no matter what. It is not dependent on situations or tasks or people. Oh, we don't have a job. When I was a student, I used to think like that too. But over the time, when I see life, everything won't matter. What matters is how happy you are. So if you are happy, things will come along, because happiness attracts happiness. So just make sure that whatever you are doing for 4 years in BIT, students especially, or teachers who are there for several years teaching, mentoring so many people, seeing people do well. I was a student too. Just be happy and keep other people happy. Don't make their lives miserable, or anyone's, your friends, your students or teachers. Just don't do those things. Just get the best out of the time, and that will take care of things. And as Vishal was saying, that things are changing, things are getting into better shape, and alumni are getting connected. We are there to help you move forward and make your name, make BIT some name as well.
Yes, Anirudh, that is what the time requires, and we are really means doing our level best as a faculty, as a management, as an alumni association. Everybody is trying their level best. Now the student has to take this particular things from this particular point of time. Ki how should we utilize this? As you were correctly mentioning, they are spending 1.5 hours of their time with us. Attendance is not there. It is not at all about attending. If you are spending time, make every second count.
That's right. And I'm surprised, how many people are there today?
Right now we are only 19, including me, you, and Surinder sir and Devashish. Otherwise, we had maximum attendance of 34 at one point of time. So,
well, I think, Vishal, it may be that technical talks are mostly very specific talks, and people might not be interested because they don't know the content, which is fine. So I would say that to maximize the impact or our time for the benefits of everyone, if we can have some topics, and I'm sure that you are doing it, I just wanted to re-articulate, have the technical talk, at the same time have something related to their career. Students get attracted by who is potential recruiter or jobs or higher studies. So you can divide the talk into two parts. Say that 15 minutes would be technical talk, and then rest of the time would be about career related jobs, companies, and then people probably, and you can bring one or two speakers at the same time. So for example, I'm an academician from academia, so 15 minutes, 20 minutes given to me, and we can bring one alumni who is working in the company, and then most people would come because they think that, oh, it might help me. I think that might be something that we can, our bottom line approach is we want to help students. It's not that we want to just say something here and walk away. If students are not coming, let's go to the bottom and what interests them, right? And then make them run the show. You be the leader from the behind. Let the student take the lead, and his responsibility is bring the whole lobby of the hostel or whole hostel to listen to the talks or interact with the talk in person.
Yes, Anirudh, I will definitely look into this suggestion also. And while your talk was going on, one thing more clicked my mind. Ki from next time, whenever a speaker is going to speak, I'll ask him or her to give me a 15-second or 30-second small clip of what he or she is going to speak about, so that if it could be circulated along, ki what is the topic actually. Because, for example, with your topic also, I circulated this hoping that this is more towards the pharmacy side. But when you started your introduction, whereby you mentioned that it's from polymer, I immediately shoot the mail to all my polymer as well as biotech as well as chemical faculty members and students to with a request to attend. So this one thing can be done again from next time.
So I had this impression that this was circulated to all BITNs.
Yeah, it has been circulated to all BITNs, all extension campuses, even through our alumni portal. It has been sent also on LinkedIn. You must have seen on LinkedIn also. So I don't make any things unturned. Means I try my level best to attract everybody.
Okay. Okay, so maybe some keywords people must attend who are related to chemicals, polymers, biotech, pharmacy, MBA student, jobs, entrepreneurs. If that keywords come, probably that would help as well.
Yes, exactly.
Well, it's okay. I mean, even in Johns Hopkins, sometimes we don't get a lot of students as audience because, somehow it has to match their interest. If that does not match, they get lost.
Anyway, I have recorded this session. I would try to put this up on our YouTube link, and I will try to circulate it to all our students to at least get the benefit. If they are missed out, they are not missing it out completely, though they miss the interaction. But otherwise, the knowledge is available for everybody.
Right. I would say that, just don't make it open for everyone. Just keep it for only BITNs who have BIT login IDs. Okay.
Yes, definitely.
Okay. All right. Anything else, students or faculties?
Dr. Singh,
Yeah.
can you give regarding TOEFL, GRE, or ILB score for your to go into your research group? How much your TOEFL, GRE, or score you need? Actually?
Oh, GRE and TOEFL score.
Yes.
Honestly, Dr. Sen, I don't know right now because it's been so many years. But I would say Hopkins, in general, biomedical engineering is number one. It's very competitive, but there are other programs, material science, chemical engineering, that can be targeted. What I want to say to everyone here through you, if you have published papers, okay, one or two papers, first author, second author, third author, does not matter. I would encourage you as a department head to involve students, even if they do one experiment or two experiments, put their name on the paper that is getting published. It is not like this that, oh, he has not done enough work, he should not get credit for the papers. It is not in USA like that. Most of the top universities, they would acknowledge their work, whatever they have done. There is no harm recognizing those people, uplifting them, because that is how they get selected for the higher step or next steps. A student here in United States, they are doing undergraduate, they don't know much about it. They have helped PhD students, postdocs, or whoever. They get names in the paper, and they get like several papers, and they get their application is higher because they have paper publications to show. In India, when I was there, I don't know about the culture right now, people won't give the names. Oh, they say, oh, you have not done anything. That attitude has to go away. Recognize them, uplift them. If you are able to make their names appear in the paper publication, their chances of getting admission into any university in US is very, very high. And you don't have to target only Hopkins. You can target, other universities. University of Maryland, that is also a very good university. So it is not the top university. Back in India, we always think about Harvard, MIT. It is not like that. It's a different game. There are so many great universities. They let them come and explore them here, and they can find a way out. What I want from the department, from the university, don't be that hard on students. Okay, we don't have that great facilities. We are improving every year. Consider that. Even if they are doing something, recognize that. Give them the platform. Okay, you have the authorship as third author, fourth author. Don't fight over it. Give them that, empowerment. If you have that, they would succeed in their life. I did not have any publication when I came out of the BIT or even masters. That was not the culture. But now we have to change it.
Some students are working also. The problem what you are pointing is very good, Saisan, because teachers they think that since he or he don't have much contribution, so why I give him or her the credit? That's why our attitude should change. A good Saisan. I'll talk to my colleague. Now some few students are working nowadays. It is not like our earlier. Now in material science and in polymer, in chemical engineering, we have good facilities. We have good facilities, but problem we could not attract students to work. There is a very big problem. Everything many students they want job, and you know that in job in India mostly software related, and very few industry. I have worked in industry 4 years, government industry as well as private industry, but I am not satisfied that what industry they are doing. Very few industries they are working on R&D work, and your research work is basically R&D work. And what you are talking about entrepreneurship is that type of research that application based. So that we have to we could not reach that level. A lot of ways there.
Definitely that our success is based on student success. So one way to get out of that situation is work on industrial related projects. So if we can bring some industrial projects and tie up with industry, students might get interested. And if students are more inclined to software job, that is fine too, no problem at all. But the point is we have to find a way to let them do what they want, but at the same time utilize them, show them their future, that what you can be like doing this, right? You can still apply your computational knowledge and skills. At the same time, you can do wet lab experiments and have some special tools that makes you very unique. So somehow we have to find a way. It's our job as teachers, I would say that, because students don't have, their vision is limited. They see jobs, money, but what about after 2 years you are getting double that money? What about after PhD you are getting 5 times of that money? They don't see those things, right? So eventually Dr. Chandan Guria was there, Dr. Sakhel was there, Goswami was there, Dr. Goswami, Mandra Mukherjee. These were, these were people who were talking to us that why should we go to higher studies. That's how I came for higher studies. Otherwise I was just looking for a job as well. So somehow there's a misconnect between teachers. There's a lot of difference between teacher and students in India. Students are so scared of teachers, and teachers are so off the road of students that they are not friends. They have to be open. They have to speak their mind. If the culture is not that, then it is not going to work. So I would definitely say that just ask them to be open and don't judge them. Just they are who they are, and even if
now projects is 2 year, 1 year. On earlier in 6 months, nowadays you're one mini project and seven semester and another 10 credit, 12 credit for 8 semester. But they are reluctant to do. Main problem we have good facilities, not like when you are passed of that time no such facilities are there. But now BIT is many new IIT specifically regarding polymer, material science and chemical engineering. The facilities we are having is comparable to many IITs, many NITs also. But somehow we could not attract the students go for PhD. Very, very big, so I can say a drawback to us. I'm also trying to tell the students, but anyway, I do not know why you're main that industry will come out. Industry will give the sponsorship project. When industry give the topic, because immediately they don't give the fund. They will see that some positive things are there, then they will give the fund. It's a big problem in Indian industry. And they anyhow they do not have that, we could not reach that type of confidence so that industry can support to us, except some personal your contact. I am not talking about your ICT Mumbai or earlier UDC Mumbai. We have very good connection with industry. Even I have seen in your western part of India, IIT Gandhinagar also, the faculty there, because the industry are coming to the your institute. But in here, the location of BIT and nearby industry also, their work they are doing very preliminary. That is not a research level type of work. So one industry actually phone call to me that your some silica you want to prepare. The silica technology they are buying from your maybe south.
So
that is your.
I think we might have to do some brainstorming session with students, like bring some students who oppose to do research or who does not like to do research, and bring some students who are interested to research. And you can bring Dr. Chadda or Surinderji, Vishal, and have a brainstorming session, open session, like having different layers of people, like very experienced people like me or you and Vishal, everyone together, as a committee, or just have some meetings, like why students don't like, what they are thinking, and don't judge them. Just say that, okay, just tell me why you are thinking, what you want to do, why you are thinking that way. Different type of people, and I try to understand, and then probably with guidance of Surinderji and other people, we would be able to, at least make a map of plan that this works, this does not work, how to inspire people to do more research, or we can do something which is related to computers